Welcome students, we will be discussing about lysosomes in this class. Lysosome was discovered by Christian D. Dave in liver cells. It is found in all eukaryotic cell except some fungi like yeast and neurospora where it is absent. So it is absent in yeast and neurospora. Otherwise it is present in all eukaryotic cells. Then the number of lysosome will be more in cells in which intracellular digestion takes place. For example, WBC cells, phagocytes, okay, then osteoclast. Osteoclast are the cells of the bones which eat up the bone cells to be replaced by other new bone cells. Then the shape of lysosome is spherical, like this. It will have a spherical shape. Inside the fluid will be called as matrix, and outside it will have a membrane. There are 4 types of lysosome based on the nature of the matrix, based on the type of matrix they have. So there are 4 types. So it will be primary. First type of lysosome will be called as primary lysosome, where it will have inactive hydrolytic enzyme. So it will have inactive hydrolytic enzymes. Then there will be the secondary lysosome, in which it will have active enzymes. And then there will be the third one called as tertiary lysosome, where it is also called as residual body. In some books you will find it as residual bodies. So these are also actually lysosomes, and they will have the undigested food material inside them. So they have trapped the undigested materials inside it. And lastly, they are called as the autolysosomes or autophagosomes. Autolysosomes, or also called as autophagosomes. These are actually the suicide bags. Suicidal bags. These are the suicidal bags. So they will release the hydrolytic enzymes which will destroy its own cell. So those are the autophagosomes. Now there are 6 categories of enzymes that are found in lysosome. So enzymes are of 6 types. 6 enzymes are found in lysosomes. Now let us just know the name. What are those names? First is called as proteases, that will break down any protein. Then nucleases, that will break down nucleic acids like DNA, RNA. And then there are glycosidases, that will break down carbohydrates or polysaccharides. Then there are esterases, that will break down the esters. Then there are phosphatases, and then there are sulfatases. All these are hydrolytic enzymes, and they are all active at acidic pH. Okay, so all of them, they will require acidic pH. And if we talk about the function of lysosome, it will be to digest food from outside. So functions of lysosome we can write as heterophagy. Heterophagy, which means digesting food from outside. Then autophagy, that is digestion of reserved food. One's own food that is reserved can be digested. Then there is autolysis. breaking down of its own cells or different organelles of the cell will be called as autolysis. Then they also destroy carcinogens. Destroy carcinogens. Carcinogens are substances which cause cancer, and they are important as defense. As a defense organ, they are very important in the cell. So now that we have understood about the lysosomes, our next topic, let us discuss about the vacuoles. So for that I need to rub the board. So children, we will be talking about vacuoles now. Now vacuoles, as you know, they are also originating from the Golgi bodies. So after the Golgi bodies has finished preparing all the final products by modification and packaging inside it, the lysosome as well as the vacuoles are prepared. So our next topic is vacuoles. Now the vacuoles, if you look, they are also spherical like this, and inside it also they will have matrix, and they have a membrane which is a single unit membrane. Okay, there is only one layer. We know that all the plasma membrane, everything, they will have double layered membrane, but here it is a unit layered membrane. membrane. So it is a unit membrane. You can write it in fact. Then in animals, the vacuoles will be small in size, but there will be more number of vacuoles, and in plant cell, it will have a large, one single large vacuole is found in many of the plant cells. This is absent in meristematic cell, in bacteria and mycoplasma. Okay, so it will be absent in bacteria, mycoplasma and meristematic cells. In plant, it will function to store water. So it will be filled with sap, we can say, filled with sap in water, in plants. Sap here means water plus mineral salt. Water, mineral salts or sugars, whatever is can be dissolved. Okay. Yes, ma'am. Good morning, ma'am. Good morning. Good morning, ma'am. Yes, yes, ma'am. Yes, yes, yes. I'm monitoring, and because in my house also class is going on, so I am able to see who are attending all the classes. Yes, yes, ma'am. No problem. Thank you, ma'am. So, students, uh, this vacuoles, as we know, the function of the vacuole in the plant cell is that it will be filled up with sap, which is the water and the mineral salts and sugar present. So all together mixed will be called as the sap. And the vacuole will also function as a storage organ. Then it will also help in cell elongation. That is changing the shape of the cell. It will help in cell elongation. and it will also help in the Turgor process, that is maintenance of water inside the cell, maintenance of shape and size of the cell will be all taken care by the vacuole. Then there are few different types of vacuoles. Some vacuoles are called as food vacuoles. So some will store solid food like phagosomes, and some will store liquid foods called as spinosomes. And there are some gas vacuoles which will collect the gases that are excreted out, and there are contractile vacuoles. As in amoeba, we see they have contractile vacuoles. These contractile vacuoles will collect the extra water that enters into the cell, and they will collect that water, and they will release the water outside the cell. So it is mostly observed in amoeba paramecium, that is the protozoans. So thus helping in osmoregulation, then excretion. So this was about the vacuoles. So our next topic after that will be cell inclusions. So the next, before I take up next topic, I'm just rubbing the board. So cell inclusions is the next topic. This include all non-living substances inside the cell. So all non-living particles, molecules that are present inside the cell. This will include food such as starch grain. For example, in potato, liver, or in the muscles of animal, we find that starch grains are stored. Then as glycogen granules in the liver cells. Then as proteins, some proteins that are floating in the cytoplasm, like allurone protein in maize. Then there may be certain oil droplets. For example, in adipocytes, we see in adipocytes those large oil droplets are present in the cytoplasm. So those are also actually cell inclusions. Then we will find certain inorganic substances such as silica. In grass, we find a lot of silica presence in the as cell inclusion within the cell, especially the epidermal cell will have silica in it. And then there can be calcium carbonate. In the epidermal cell of bitter gourd, we find calcium carbonate is present. Also calcium oxalate. Water hyacinth has got calcium oxalate in its cells. And then there are certain gases like oxygen, carbon dioxide, nitrogen. They are also present as cell inclusions. Then waste material. Waste materials or waste substances such as ammonia, urea, and uric acid. So all these are the cell inclusions. After the cell inclusions, let us now discuss about the microbodies. So for that, again, I need to rub the board. So our next topic will be the microbodies. They are found in eukaryotic cells. and they arise from endoplasmic reticulum. So they are made out of endoplasmic reticulum. There can be several types, such as, let me just give you the name. So one is spherosomes. They are found only in plant. Okay, so they are found only in plants, and they are also called as plant lysosome. Okay, they act like lysosomes inside the plant. Then next is glyoxysome. Glyoxysome. This is also found in plant only. Okay, and they help in oxidation of fats. So oxidation of fat in the plants, in the plant cell. Okay, then there is peroxisome. It is found in both plants and animals. So both plant and animal cells will have it. Especially it is found in liver and kidney cells. It helps in the formation and breakdown of hydrogen peroxides and is responsible for photorespiration. So it is responsible for photorespiration. We will discuss about photorespiration in the coming chapters. Okay, we have a chapter of respiration in which we will discuss about it under the plant physiology. So with this, the chapter number, this cell, the unit of life, this chapter is over. Thank you, students. Please repeat the video again and again, and you can study from here.